Everybody smile. I do this every year at this meeting because I'm always amazed at the numbers of people that show up to hear me talk. All right. So as Michael said, I direct CIFAS, and hopefully most of you are familiar with what CIFAS is, but I'll give you kind of a little background, hopefully. So NIAAA, National Institute on Alcohol Abuse and Alcoholism, established and funded the collaborative initiative on FASD, or CIFAS, in 2003. And it was a consortium that was designed to inform and deal with effective diagnosis, screening, interventions, and treatment approaches to FAS. And they wanted highly integrated projects, and I'll get to that in a minute of what they were trying to do, with multidisciplinary research approach, looking at teams of both basic scientists and clinicians, and trying to look at various cohorts at different countries. So it grew out of two international meetings that were held in France in 1997 and then Spain in 2001. And these grew out of the fact that there were advantages in doing large international multidisciplinary collaborative projects. At that time, single sites really didn't have enough subjects that you could answer questions with relative ease because you didn't have enough subjects. So there were sites around South Africa, in Russia, that had larger numbers of people who were impacted by prenatal alcohol. And so the single site created a problem. Like San Diego, I think we had 25 kids in the first couple of years. The other thing about the sites is if your clinical findings are similar across sites with varying environmental factors or confounding factors, it suggested it's due to the alcohol exposure. If they had different effects at each site, then there was something environmental going on that might give you ideas of mitigating or resiliency factors. And the main thing was that they recognized that basic scientists needed to talk to clinical scientists and that we all needed to talk to stakeholders. So CIFAS has gone through 5 iterations. Each has been roughly 5 years long. New people come in and out of CIFAS every 5 years, or even more frequently than that. We try to turn over occasionally so we don't have the same people doing the same things for too long. Each of the versions builds upon the preceding version. It's in its 21st funded year, and ever since its inception, we tried to involve stakeholders. I mean, I don't know how many people here in Seattle remember the Pest of the West. There were 3 women here who bombarded the legislature for funding for fetal alcohol, and they were on our first advisory committee for CIFAS. So the focus is to enhance the diagnosis of FASD at different stages of the lifespan based upon biological, physical, and or behavioral assessments, and to improve the outcomes. They want to build capacity using e-health technology, and that's a current aim of CIFAS 5, and you'll hear more about that from the other speakers who are going to be talking about the research going on at CIFAS. All right, so I don't know how well you can see those little dots up there, but that's the sites that CIFAS has had projects in. You know, we're well represented in the US and Canada, in Europe, and we had a project in Australia and one in South Africa, or a couple in South Africa. So we've been all over. Basic organization, as I said, there's clinical science projects and basic science. And this is for the current CIFAS. There's an administrative core that I direct. There's a data coordination center that Leo Weatherville does, and a dysmorphology, dysmorphology that Miguel de Campo, and some of those people will be speaking you. And then we have these science projects that interact with each other. And then we also have some UH2 projects, which are high-risk developmental projects. We're advised by NIAAA, in particular right now, Peter Dunte and Elizabeth Powell. And we have a science advisory board of eminent group of individuals with various expertise who kind of give us and advise us on where we're going, what steps we should be taking, et cetera. So the consortium kind of looks like this. Myself, Michael Charness is the scientific director. Jennifer Thomas is our administrative specialist, and she deals a lot with the web pages and things like that. And then Jill Vanderbilt, who runs the whole show. I couldn't deal without her. And Science Advisory Board and all of the PIs on the project are part of the steering committee and our NIAAA advisors. Now, the admin core also does some things that utilizes a lot of outside help. So we have outreach, which is done by FASD United, and Tom Donaldson is a PI on our, within the admin core. He's on a subcontract. We develop apps and e-health technologies with Blue Residents, which are done by Gantz. And then we have dissemination and implementation, and this is a new aim of CIFAS, is to try to get our results, the things that we're doing, out there for people to use on a wide scale. Here's our conceptual framework, and this has been it since the start of CIFAS. Is that prenatal alcohol exposure, depending upon the dose, timing, pattern, genetics, et cetera, impacts embryonic and fetal development. That has an impact on brain, that has an impact on behavior. The brain and the face seem to go somewhat hand in hand, and the behavioral effects resulting from the brain can be modified by environmental or genetic factors. And over the years, we've added other things. We're now interested in FASD over the lifespan. It used to be we studied mainly kids. Now we're studying from all the way up to 60, 70 years of age. We're interested in long-term health consequences, as you'll hear. That's become a major focus of CIFAS. We're interested in interventions and in screening. We're trying to meet capacity demands. There are many more people impacted by prenatal alcohol exposure than service providers. So how do we deal with that difference in the numbers? We also this year, or a couple of years ago, added a prevention project. And again, we do outreach, implementation, and e-health. I said it's collaborative, and this is just the interactions that occur between the various projects. That's one of the things that you have to do. If you come into CIFAS, you just don't have an individual project. Your project has to line up and match up and be able to interact with the other projects in CIFAS. And so we have this chart that comes out, and this shows you how well people are collaborating with each other and who's drawing blood at what site and where that blood goes to, saliva, who's doing what. Jeff Wozniak's doing similar things to Sarah Matson. So we keep track of that. We have monthly meetings to discuss all of this stuff, and we invite outside speakers to come and talk to us. And that's, we have an annual meeting as well, and we have, after this meeting, we're having one of our 2 annual meetings. Now, CIFAS has been, I think, fairly productive. We've passed the 400 publication mark. This is just from CIFAS 5, are the lines in purple. But you see, we publish in some very high-quality journals, Lancet, Nature Reviews. So we've had some fairly big impacts in the FASD field, publication-wise. We've recruited a lot of people into the FASD field. We have people who never even thought of doing it because we could give them a little bit of money to start up, get people involved, or get them involved. This is a very interesting field, and they stay. And so we've done been very successful in recruiting and keeping people in the field. We have a website that you can go to. We have a bunch of things on there that, resources, et cetera, et cetera, et cetera. But I want to point out our data sharing. All of the data that's collected in CIFAS is made available to anybody who wants it, who can meet certain requirements. So if you're interested in anything that you hear about or we do, you should look at our data sharing. You may want to get some ideas to use our data to answer your questions. And this is just what's available. Right, I said there were 5 iterations of CIFASD. The data from the first 4 are already available online. And there's things such as dysmorphology, behavioral assessments, all types. And there's, I guess, over 1,000 individuals represented, but we don't have data on every category for every subject. We have a pretty good research visibility. You know, we are at the RSA meeting every year. We've got a symposium coming up, or we had one. We have another one coming up. The members talk. CIFASD people like to go out and give talks. And a lot of presentations. Our research visibility, we were at UFAS again last year. We're doing we did several oral presentations there and poster presentations. So we like to get our word out about what we're doing and attract other people into maybe thinking about applying for a CIFASD project at the next renewal. Just more examples of talks and how our investigators are involved in both regional, national, and international meetings. Our outreach and education support. CIFASD investigators are encouraged to get out there, get in the local scene as well. Red Shoes Day, we all put on red shoes and walk around, and Christy Petrenko has a big run that she does that day. Consortium members get in the media quite a bit. Claire Coles was on PBS. Jeff Wozniak has done a podcast. They get good press, so I think we're getting information about FASD out there and the latest research that's going on. And we tend to be involved in projects that have somewhat of a risk, but maybe a higher payoff than most normal R01 type applications. This is the investigators that we currently have in CIFASD. And I just want to say, if you want more information, we have the data sharing that you should look at to see. We're certainly happy to consider your project. And then for overall CIFASD and participation, if you see studies that you think you might be able to contribute to, you know, say something to one of the investigators. I mean, we're always happy to collaborate with people. And I think I'll be finished.
Thank you. Our next speaker is Amanda Mahnke from Texas A&M College of Medicine. Mrs. Ford.
Okay, perfect. Good morning. I'm excited to show you guys some data that's actually been building across multiple CIFASD iterations about these little tiny RNAs that we can find in the blood called microRNAs, and how we think they might have this interesting role in being able to indicate risk for adverse outcomes. And so this is not the room where I need to say this, but I'll say it anyway. We need additional diagnostic capacity to diagnose fetal alcohol spectrum disorders. And so what we're thinking about is what biomolecules could do that. And so we have things like ethanol metabolites that do a really good job of saying, hey, prenatal alcohol exposure occurred, but they don't talk about those risk and resiliency factors that are present in the children that determine how bad the outcomes are gonna be. So we're thinking about, can we find biomolecules that can reflect that risk and resiliency and stratify the kids who have been exposed to figure out which ones are more likely to have adverse outcomes. And so we think microRNAs, my favorite non-coding RNA, are the ones who can do this role. So if we think about this big term called the central dogma of molecular biology, all it's saying is that DNA goes to RNA goes to protein. And microRNAs are right in between that RNA to protein step. So they block the production of proteins. And so these microRNAs don't just hang out in a cell. They can actually be released from the cell and end up in circulation. And because there's a lot of microRNAs in the body and they're related to biological processes, we think that when they're released, they provide a systemic snapshot of both adaptions and maladaptions in development and what's going on in the organ systems. And then I'm also going to show you in a second that we don't just think they're reflecting changes in the cells. We think they might actually also be mediating issues in the cells, including following prenatal alcohol exposure. So the data I'm showing you was collected from a large prospective cohort in Ukraine over many, many years. In this cohort, pregnant women and their children were recruited. I'm going to show you some data from the first publication that just looked at maternal plasma samples from the second and third trimester. I'm going to show you some data from child plasma samples that were collected from children at about 3 years of age. And then I'm going to show you some data where we have a small subset in here where it's moms and their own infants. So it's maternal child dyads. They're paired together. And so you're going to see me talk a lot about PAE status, so whether or not there was heavy alcohol exposure during pregnancy, as well as neurodevelopment status, so typical and atypical, which was assessed by Bayley scales of infant development. So me as a basic scientist, we got these plasma samples, and I got to sit there in the lab and pull out all these little microRNAs, and we ran them on qPCR panels. We love this technology. This is the same technology that was used for overnight COVID tests, so there's some really good potential here. This is in the clinic. And so we looked at 752 unique microRNAs. And so first of all, the first study we looked at where we looked at those microRNA biomarkers in the moms. So this is while they're pregnant. And in this study, we divided it into 3 groups. We have an unexposed control group, and then we divided those pregnancies with heavy alcohol exposure in 2 groups, depending on infant outcome. So if the infants showed growth deficits and neurodevelopmental delay, those pregnancies were in the HEA group, where it was alcohol exposed, infant affected. And those where there was alcohol exposure, but the infant did not show any hallmarks of FASD, that was heavy alcohol exposure, apparently unaffected. And so what we found is this group of microRNAs that are shown by the red color in this heat map that were elevated starting at that second trimester in that HEA group with the affected children, not only from the unexposed controls, but also from those pregnancies with similar levels of alcohol exposure, but those kids were resilient and didn't show signs of FASD. And so we called these the HEA microRNAs. And so then we had a talented MD PhD student in the lab who said, well, what are these microRNAs doing? Can they do something on their own? And so he took these HEA microRNAs and went into a mouse model and administered just the microRNAs by themselves and found that the microRNAs were sufficient to significantly decrease fetal weight and significantly decrease placental weight, indicating that these microRNAs are not just indicators, but also potentially mediators of these growth deficits. And work is ongoing looking at placental involvement for those microRNAs and how they act on the placenta. So then we also went into the child samples and said, well, what might microRNAs be telling us in the children? Do they have the same explanatory power as in the moms? Do they show us something new about the children? And so shown are volcano plots. The up and out is the interesting part. You can see that they're colored. And so hopefully you can appreciate that by about 3 years of age, we're not finding a lot of microRNAs associated with prenatal alcohol exposure status. That happened in the back. Okay, it happened. But we are finding a lot of microRNAs associated with neurodevelopment status, indicating that these microRNAs are reflecting these ongoing changes in biology that may indicate risk for adverse outcomes in the children. And so I want to show you some new data from a talented grad student who just joined my lab. And you can tell how new he is because I said, hey, Darius, there's this thing we use in RNA-seq when we're looking at 10,000, 12,000 genes, and it's called weighted gene co-expression network analysis. Yeah, go look at those microRNAs and try and apply it to 140 of them. Just go, it's fine, it'll work. I didn't think it was gonna work, but he got in there, dug it apart, and it's been working. So shown on the left is a heat map. And so the key features here are these hot spots are places where microRNAs are moving together across the samples. And we can divide them into little groups, and since they're kind of hanging together, we think they might be acting in a network and acting together. And then we can make this lovely constellation graph and look at their relationships, and it's beautiful. I think it looks like, I guess this is my new sign. I'm no longer at cancer, I'm a microRNA map. But so we wanted to know, in these new networks that Dry's discovered, what might they be telling us about the child biology? And so we looked at what pathways might these microRNAs target. And so here, the purple colors are you can see the 4 modules on the top. Ooh, does that work? Okay, so here are the 4 modules that Dry has identified. Here's some pathways, toxicology functions that were indicated as being targets of those microRNAs. And the really cool thing is, the first question I always get asked in Q&A, so you don't need to answer it, is where are these microRNAs coming from? And this is the first time that we've started seeing clustering by organ systems. So we have a group up here that's associated with liver, a group right here that's associated with kidney, and that's a lot of that of a 4th network, and then a group right here associated with cardiac. So you better believe I'm going to go talk to Olivia and see if these microRNAs are targeting some of her interesting cardiac genes. And so, because this is such a rich data set, we went and looked in at those 42 maternal child dyads, so moms and their own children. We found 145 microRNAs that were expressed both in the mom and child samples, and those are what we looked at. And so our first question was, are there some maternal child microRNAs that are kind of hanging together in the way that they're changed, indicating that there might have been some biology that was present in the mom that you also see in the child? And so these are barbell graphs. The really important components of this is how close are the 2 dots together, and then also we're really interested in this white space. The dots in the white spaces are likely clinically relevant. And so you can see on the PAE status side that there's not a lot of these balls that stick together. There's not a lot of maternal, shown in maroon, microRNAs that are expressed or similarly have differential expression by PAE status as in the children. There's a couple, but not a ton. We see a lot more by neurodevelopment status. There's some microRNAs that seem to be moving in the same direction when you stratify by neurodevelopment status of the child, both in the maternal and the child samples. And so we were wondering what biology this might be telling us about. And we were surprised that we had this really big signal of collagen synthesis and extracellular matrix pathways. That's not necessarily where we thought those were going to be pointing, but definitely is worth a little bit, some more investigation. And so we also wanted to look at, we have maternal and child data. You could easily think that, well, the maternal data should show PAE status, and the child data should show neuro status. That's them, that's their data. And we found that if we try and figure out what variables most indicate whether you have PAE exposure or not, and so this is a random forest analysis. It's these top guys that are the most predictive of if you're exposed or not. It's maroon, it's almost all maternal samples. Well, that makes sense. There was the direct exposure. However, when we get to neurodevelopment status, we see that there's some child microRNAs and some maternal microRNAs. And interestingly, no maternal clinical variables that can indicate atypical versus typical neurodevelopment. And when we use something to look at the core group of microRNAs here that might have the best prediction, we get a model that's going to make Sarah Matson's data look really good. So we get a model. So this number right here says our model's performing okay. We think of a model of 0.7 and higher as maybe usable in the clinic. So we're real close. Again, you're going to see Sarah's and it's going to be impeccable. But for us, we were thinking, what might be a key variable we were missing here that could be important and help us improve the predictability of some of these microRNAs? And so we looked back at some of our data that was looking at contributions of fetal and child sex in maternal analysis alone and child analysis alone. And so it's a measure of cross correlations of microRNAs. The important thing is we're looking how the mountains move. And you can see in the 3rd trimester, both in the males and females, in the HEA group, there's an increase in correlation. So the males and females are looking similar. However, in the 2nd trimester, the females start showing that signature early, and in that HEA-UA group, we see this interesting increase that seems to resolve. So in the 2nd trimester, we're seeing maternal microRNAs changed in an interesting way based on the female fetus, so the sex of the fetus. Then also in the child samples, when we stratified by sex and then looked at PAE status, we found similar numbers of microRNAs associated with changed in males and females by PAE status. However, we found this really interesting phenomenon where there's a lot more female microRNAs that were altered by neurodevelopment status. And so we were thinking back to this dyad pool that doesn't have all these subjects, it's a little bit smaller. Do we see sex differences in that? And I'm going to show you guys the maternal barbell graphs from those models. So when we look at maternal, the maternal microRNAs, and we look at comparing male to female. So on the left, these are all of the microRNAs. These are all of the microRNAs that are changed to a clinically relevant effect size by neurostatus in males. We get 42. And so then we go to the females. And there's a few more microRNAs there. We see that the females have a lot more microRNAs that are changed by neurodevelopment status. So what does this mean for our predictive models? Well, if we think about those microRNAs that were in that neurodevelopment status model that had both child and maternal microRNAs, so we have a little bit of a core signature that could be predictive in both male and female children, and in pregnancies with male and female fetuses, but then we have a big signature that's sex-specific. So we might be finding that we actually maybe have a model that would perform better in female children. And we need to think about, if we take this into the clinic as a diagnostic tool, we likely have a core group of microRNAs that can be used both in males and females, but if we want to increase our specificity, we need to also add in a couple of specific female microRNAs for the female panel and a couple of specific male microRNAs for the male panel. And so we're investigating into that a little bit more. And so with that, hopefully you are convinced that the microRNAs are cool and could be potential biomarkers of impacts following prenatal alcohol exposure. I hope that you also thought these microRNA networks were interesting, and I particularly find them interesting as indicating tissue sources. We found a model for atypical neurodevelopment that contained both child and maternal microRNAs. However, it looks like biological sex is an important mediator of microRNA expression, and we need to consider this when we think about taking this into the clinic and thinking about its utility there. And with that, I'd like to thank my lab members and some members of our collaborator, the Miranda Lab, who are either actively contributed this data or are continuing with some ongoing work with this data. Collaborators outside A&M, our funding sources, and then we have some really great organizations in the state of Texas, including the Texas FASD network. You've probably run into a couple of members all throughout this conference already. And so I wanted to shout out that Texas is trying to do some good work and increase the knowledge of FASD. And with that, I would love to take any questions.
Hi, Amanda, great question. So you showed that there were some sex-specific microRNAs for males and females. Have you mapped those back onto the genome to see whether they fall on particular chromosomes or sex chromosomes, or are they scattered everywhere?
They're scattered. I mean, the first thing you go is the X chromosome has a lot of microRNAs hanging out on it, and very few of these are X chromosome located. So the source is still a little bit unknown as to why we're seeing a sex difference in pattern. And what's interesting, though, is Mike Suddy also finds sex differences in facial dysmorphology. He's here somewhere. But Sarah doesn't find any in behavior. And so there's this interesting thing where there might be two biological routes to get to the same neurobehavioral outcomes that Sarah's seeing. But yeah, that's a great question. We're trying to figure it out.
Thank you for the talk. My head hurts a little bit after basic science clinician sometimes, but the bit about collagen interested me.
Yeah.
There's a lot of work especially a colleague in the UK is doing hypermobility syndromes in neurodevelopmental disorders, and collagen is part of that. And we know that collagen is a scaffolding part of the brain. What's the relationship do you have with the collagen bit of here, the story, and alcohol in there, and is there any interrelationship?
We don't know a ton yet. We were not expecting collagen to actually come up as well, I wasn't. Others might have been, but I wasn't. But Roger, you bring up a good point. Like we need to follow that up. Especially, I need to talk to some of the experts here, such as the ALC, about joint mobility and hypermobility, and start thinking about that. But it was also interesting that we saw it in the mom. So maybe it's going after the same collagen pathways, but maybe in the mom it's placenta related, and then in the kids it's Ehlers-Danlos or another connective tissue disorder. But that's a great point. And I would love to talk to clinical people who can help me understand that a little better.
Good morning. Great presentation.
Thank you.
What I'm learning and going to the various research sessions is that there seems to be some differences between the sexes over time. And there was this longitudinal study done from Berkeley about ADHD in females compared to males, and that it's more typical for males to prune the symptoms of ADHD during adolescence. But when females are diagnosed with ADHD, it's a lifelong issue. So I'm wondering if, the research, if there's correlation between looking at for all of the people that are looking at this, the sex-based differences, because the typical first diagnosis for people with, FASDs is ADHD. And so I'm just curious about that, as a marker, and then longitudinal studies over time of the impact of not just the functional or the behavioral adaptive living skills, but perhaps even as people with FASD age, the physical problems that show up later, post-adolescence.
Yeah, that's a great point, and something we see time and time again with a lot of neurodevelopmental conditions is that we think it occurs less in females because they don't act out, they don't have these weird behaviors, they mask a lot, and so that also might be something that we need to consider in terms of diagnosing and getting kids who will end up being diagnosed with FASD in the clinic. So that's an incredible point. The great thing is CIFASD is really doing good work and looking at sex differences, but we need to keep that in mind. You're 100% correct. There may be different risks there by sex. There may be different presentations we need to consider, or how ADHD comes in and what it looks like, and that might be different as well. You're correct. Thank you for that point.
Hello. I'm Yasmin Centurious. I'm a developmental behavioral pediatrician, so completely clinical. So forgive me if I am asking from a purely clinical lens. We do whole exome sequencing in a lot of our children. So I see children, I diagnose them with FASD, but I also do genetic testing to make sure we swab and then send out. Is somebody looking at how those whole exome sequencing variants or pathogenic results actually correlate to some of the mRNAs? Because I think we're seeing quite a lot of these, and sometimes the expression of FASD is different depending on whatever genes there are that we identify.
Yeah, that's an excellent point. And one of the great things about some of these samples is Tina Chambers at UCSD has some genetic data set. It's just not ready for us quite yet to mine into, but there's been some great data from Susan Smith looking at it and others. But that's a really good point. There could be a genetic source for this. It may not all just be environmental response. That's a great point.
Thanks.
Yeah.
Thank you. Our next speaker is Claire Coles from Emory University School of Medicine.
Thank you. Okay, so.
Button goes forward.
Hi, good morning. Nice to see all of you. Yesterday, we were going to talk about the, this program, I was supposed to talk about the medical or health-related outcomes. And then Susan Stoner yesterday talked about the health-related outcomes from our project, so I thought maybe that wouldn't be the best thing to do. So I'm going to talk about the impact of health problems associated with prenatal alcohol exposure on the adult's experience of disability. And the reason for, and which button do I push to? The green. Okay, to advance the slides.
The big fat green one.
Mm-hmm. This one?
Yes.
Okay. I do want to note that I have no conflicts of interest, and that all of what I'm talking about is coming from this particular aspect of CIFAS, which is currently called the multi-site study of prenatal alcohol exposure and its effects on inflammation and endocrine dysfunction in adulthood. And I and Tamara Bodner are the multi-PIs. The study is being carried out in Atlanta, Seattle, and in Calgary at the moment. And again, the whole problem that we approached was, despite the prevalence of FASD and its lifelong course, there really is limited empirical research about adult health, physical characteristics, neurobehavioral, or adaptive function as individuals get older. We have been working on this for 6 years, and as a result have been able to collect a lot of different data about the individuals that we've been working with who come from these 3 cohorts, and published a number of papers on various aspects, including a recent paper that discussed the specific health-related issues that these individuals reported. And one of the aspects that, one thing we asked was, overall, do you think yourself in good health, both in terms of physical and mental health? And in both cases, both for physical and mental health, individuals who were alcohol exposed or who had been diagnosed with a FASD replied that they did not consider themselves to be as healthy as did a contrast group, unsurprisingly, but unsurprisingly now, it wasn't necessarily so when we first asked about it. So one of the questions that we were interested in as we began this was, what was the relationship of prenatal alcohol exposure to the experience of disability? And the reason that this is an important question is that this has a great deal to do with both the individual's understanding of themselves, but also their access to services. And it is important, I think, for us to establish that prenatal alcohol exposure can make someone vulnerable to disability in various areas, and that should provide them with the access to needed support systems. So the questions I'm addressing here are, should fetal alcohol spectrum disorder be considered as a disability and entitle the affected individual to services? Is elevated disability associated with prenatal alcohol exposure as well as with FAS, fetal alcohol spectrum disorder? So talking here about the range of problems associated with it. And are there specific domains that are more or less affected in those with prenatal alcohol exposure? And so how do we define disability? Well, this is the legal definition in this country. It may vary for different countries, but basically it implies that there's a physical or mental impairment that substantially limits one or more major life activities of the individual. And I've listed a number of the major life activities as defined by the law that refer to everything from a basic ability to care for oneself physically, and things like walking and standing, to more wide-ranging activities in the community. In order to measure this, we selected a measure that has been created by the World Health Organization, and they developed a measure that was designed to measure this broadly and that could be applied from one condition to another so that there would be a general understanding of this. And this is sort of a generic instrument. It assesses function in 6 domains and provides a total health-related disability score. It describes 6 constructs. One is cognition, a second mobility, and self-care, getting along with others, which is an important issue, life activities, and participation in the community. And it looks something like this. In the last 30 days, how much difficulty have you had in these activities as a result of your particular disorder, whatever it is? So understanding and communication, getting around, physical self-care. And this is getting along with other people, specific life activities, and general participation in society. You'll note that one of the questions is living with dignity, and that refers to the issue of stigma that one might encounter in the community as well. So I think that reflects some of the questions that we've been having here at this meeting. So who participated in this particular study for us? 2 cohorts of individuals were participating. Those at Atlanta was at Emory University, and those in Seattle at the University of Washington. Those in Atlanta were selected from a cohort of individuals who had been identified prenatally, which is by their mother's report before they were born. They were predominantly African American and predominantly disadvantaged. They were born between 1980 and 1986, and at the same time these individuals were identified, a group of individuals were identified who, as contrast group under the same circumstances, and have been followed ever since. In the Seattle cohort, it is a clinical cohort who were identified at the University of Washington Clinic as having alcohol effects, either as having FAS or what was called FAE at that time, or fetal alcohol effects. We would now call them FASD. These individuals were predominantly white and Native American. They were of mixed socioeconomic status, and the controls were recruited on an ongoing basis for research purposes. Here are the characteristics of these individuals. I want you to note that for their age, their percentage of individuals who are male and female, this is percent male, and in terms of their racial grouping, those individuals who were considered to be fetal alcohol syndrome or dysmorphic, those people who were alcohol exposed, and those people who were in the contrast groups were not different. They were different in terms of the percent who were married or partnered, and we find that individuals who are alcohol affected are much less likely to have a life partner than those people who are in the contrast group. Socioeconomic status was also affected, and I think it is important to note that when the samples were selected, there was no difference in socioeconomic status, that the socioeconomic status has changed over the years as a result of what I assume to be the disability status of the individuals. And number of biological children is not different. This is the score from the HUDAS, and it is a summary score with both prenatal alcohol-exposed groups being significantly higher in terms of their reported self-understood disability status in relation to the control group. So there's a significant difference, but it is both of these groups, it is not just those who meet criteria for fetal alcohol syndrome. These are the areas of elevated disability reported by these adults, and you will note that it is not in every group that we are seeing these reports. It is in the ability to communicate, self-care, getting along with others, and the social participation in the community in which people are identifying themselves as having problems in these areas. So specifically, people with prenatal alcohol exposure say that they have significantly more difficulties coping with the effects of their exposure in understanding and communication. And the specific areas are concentration, being able to remember things, specific problem solving, and learning, understanding what others are trying to convey to them, and in maintaining conversations with other people. They also say that they have significantly more difficulties with self-care, just taking care of themselves on a daily basis. They report that they have more difficulties coping with the effects of their exposure and getting along with other people, dealing with people when they meet strangers, maintaining positive relationships with friends and family, making new friends, and also in sexual and romantic relationships. People with PAE say they have significantly more difficulties coping with the effects of their exposure in terms of general social participation. They report problems with participating in community activities, with coping with the barriers and hindrances in the world that come up, coping with stigma. They have to spend extra time in accomplishing daily activities. They have more problems coping with emotional impact of their limitations that they experience as a result of their disorder. They find their problems to be a financial drain. And they also report that because of their condition, they are having to rely more on family and others to help them get through things. So should FASD then be considered a disability and entitle affected individuals to support? Clearly, adults with PAE are more likely to report problems with aspects of daily living consistent with having a disability than non-exposed adults. The question arises whether the problems noted substantially limit their daily life, and this is going to be an individual question, not something you can assign to the entire group. Whether services are appropriate will depend on the degree of limitations and the individual's context. Is an elevated disability associated with FASD or PAE, as well as with people who have the full syndrome? Apparently so, according to the individuals who reported in this study. Both individuals meeting the criteria for FAS and those who are alcohol exposed without physical effects report similar levels of problems. This may seem somewhat counterintuitive, but it reflects previous research which has suggested that those who are so-called less affected may also have significant problems in social and emotional functioning. Finally, are there specific domains that are more or less affected? Yes, it appears that among those in this study, anyway, few problems were reported in daily physical activities and basic household care. In contrast, there were problems with cognition, problem solving, social interaction, and some of the problems were less obvious than those in other kinds of disabilities. And because of this, I feel that often we ignore these issues and attribute them to the characteristics of the individual rather than thinking of them as something associated with their disorder. And here are collaborators. I want to thank everyone here from the Department of Psychiatry at Emory University, the Department of Psychiatry and Behavioral Sciences, the University of Washington, as well as collaborators, a number of others who work with us, including individuals in University of British Columbia and Calgary. Thank you. Anybody want to say anything? No?
Claire, this really rings true with what we see in the real world, and these were real world people who participated in this. The old dictum was also that sometimes PAE was worse than FASD in the sense of worse, more difficult outcomes. And I wonder, did you look at that group and see those who had cognitive scores that would be recognized in the intellectual disability and be eligible for community living supports? Were they doing better? So the irony of having lower cognitive scores, you actually had more support for those elements. I mean, they're in a sense more protected, not only by their family, but the community around them, as opposed to people missing and the exclusion that they feel in their daily interactions that put them in chronic stress. And that's what I experienced with not only the adults I followed, but also some of the foster kids that I cared for, that the more invisible they were, the more they struggle.
Yeah, certainly this is true when these individuals are younger. They're now, the late 30s in this sample, and then now in their 40s. What I'm finding is that their caregivers and the families are often dying, or they're not there, which is causing a lot of more problems for some of these individuals, in both groups. But overall, yes, there's sort of a hidden disability in some individuals that has not been identified, and they are therefore not getting some of the care or support that they should have.
But you're saying that, are you saying that both caregivers for those who had full FAS? Because I'm just wondering, is there a difference between the full FAS, which often had lower cognitive scores? When I think of Ann's data or the Seattle group, I don't know yours. Or is it both, that when the caregivers are gone, both FAS and FA?
Well, those who didn't have the lower IQs tended not to be need caregivers or have that set up that way.
They might have needed them, but they didn't.
Yeah, and so they have a tendency to get more social troubles, I would say, more likely to be arrested and things of that sort.
Okay. Thanks.
Thank you. Our last speaker for this part of our symposium is Blake Gimbel from the Ohio State University School of Medicine.
Thank you all. I'm glad to be here. We're going to shift gears a little bit and talk about brain imaging in kids with FASD and how our team is using some of these novel tools that are being developed to understand some of the brain-based differences that we see when we work with these kids. So I have no conflicts of interest to disclose, unfortunately. A few learning objectives for today. So we'll be talking about some of the brain-based differences in thinking and learning skills and how those relate to brain structure in kids with FASD. We'll also talk about some of these novel tools that we're using that can really sort of complement some of the research that's been done to date and really gets at the ways that some of these structural brain differences might relate to those thinking skills. So first, a little bit of background. As we've heard about today, FASDs are associated with complex needs. So we know about this in kids, and we've just heard from Claire about how we see that across the entire lifespan as well. And some of these novel tools that have been used to utilize brain imaging might be really useful in a number of ways. They can provide further evidence of even subtle differences in brain development that are related to important functional outcomes that can support sort of the ongoing advocacy work and education that's happening right now. They can also potentially inform some of our intervention work. So when we get a better sense of some of these differences in the way the brain is developing over time, we might be able to target our interventions at those specific critical windows of brain development. They might also help us understand outcomes from our treatment studies. So this is something that we're really interested in exploring, whether the benefits that we see from some of our neurodevelopmental interventions might actually map onto some of these structural anomalies that we see. So first of all, I'm going to talk about how our team is using an approach that looks at brain growth charts to kind of quantify brain anomalies in this population. So many people here might be familiar with the idea of growth charts. If you took your child to a pediatrician, they would do things like height and weight, maybe even look at the child's head circumference. And using growth charts based on sort of what we expect for development in typical individuals, we would be able to plot sort of where an individual might fall compared to others of their same age and sex. FASDs are diagnosed based on a number of factors. This includes, as we know, the history of alcohol exposure, the neurobehavioral differences that we see. One metric that's used in that diagnosis as part of the physical evaluation is looking at head size. And the reason why we look at head size is it's used as sort of an approximation of brain volume atypicality or brain volume anomaly. However, some of the research is showing that head size is actually maybe not as sensitive to prenatal alcohol exposure, especially for older kids. So we do see in younger ages that brain volume and head size are tightly correlated. But after you get older than around the age of 7, we see that correlation tends to attenuate. We see that's not as strong of a correlation. And some of the early studies, one of the early studies that looked at this found that up to 50 to 60% of kids with prenatal alcohol exposure who have normal head size might still have smaller than expected total brain volumes. So we were interested in exploring this with this new approach using brain growth charts. So we've used a couple of different tools. One of them uses data from the entire brain. So we're looking at sort of the total brain volumes in different types of tissues. And another looks at some of these deep individual structures deep within the brain themselves, so at an individual level. And these tools are based on large numbers of typically developing individuals across a number of different neuroimaging studies. And they basically do the same thing that we can do with our growth charts for things like height and weight. They allow us to look at sort of how atypical or how typical an individual person is within our own sample compared to what we expect for development. So I'll talk about sort of the first way that we did this. We looked at the whole brain, and we had individuals who were part of our CIFASD study who came into our University of Minnesota center. They did a neurobehavioral testing. They also had a brain scan and a physical evaluation, which included that head circumference measurement. And what we were interested in with this first analysis was sort of whole brain tissue types. So we looked at total cortical gray matter, which is shown in purple, total cortical white matter, which is shown in blue, and total subcortical gray matter, which is shown in yellow. So the orange is showing ventricular volume, which you can also calculate using this method, but we weren't as interested in that in this study. So one of the first things that we saw was that, as we expected, the youth with prenatal alcohol exposure had smaller than expected brain volume percentile scores compared to others of their same age and sex. So what I'm showing here is for each of these different types of tissue in the brain, we're showing the percentile scores on the y-axis over here, and we're showing the group membership on the x-axis. And that dotted line is the 50th percentile. So values that are below that would be smaller than expected. And what we see is that youth with prenatal alcohol exposure are smaller across each of these types of tissues in the brain. We saw that this was particularly prominent group difference in total subcortical gray matter volume. And this was interesting because we know that those deep structures within the brain might actually be more sensitive to the effects of alcohol exposure than other regions in the brain. When we looked within the alcohol exposure group, what we saw is that those who had more atypical brain volumes, so brain volumes that were at or below the 10th percentile for their age, these were the ones who had more neurobehavioral challenges. So as one example, when we look at something like IQ, we see that the kids who had atypical volumes, and shown in blue, darker blue, were the ones who had lower scores on that measure, compared to those kids who had alcohol exposure but had more typical brain volumes for their age and sex. As expected, we saw that head size and brain volumes were positively correlated, so the bigger your head, the bigger your brain. But for a number of kids in our alcohol-exposed group, we saw that a typical head size, but smaller than expected brain volume. So what I'm showing in this rectangular area here on this left figure is the participants who had smaller than expected white matter volume percentiles, but normal head size, so above the 10th percentile for their age. And that was about 17% of our sample who wouldn't actually be captured by that head circumference measurement if we were using that alone. We were also curious to look at the relative sensitivity and specificity of these different tools. So sensitivity refers to the percentage of kids in our alcohol-exposed group who are accurately identified with these tools, and specificity refers to the percentage of the control participants who are accurately classified. And we can see that for the head circumference measurement, a small head size captured around 11% of our alcohol-exposed kids. Relatively higher sensitivity was seen for the brain volumes percentiles themselves. So when we look at the brain directly, we see that sensitivity is a bit higher, so it's around 35%. And we don't sacrifice much in terms of specificity. So most of our control participants are showing typical head sizes and also typical brain volumes. So what these data suggest is that these brain volume percentile scores might complement that traditional approach of using head size as sort of an approximation of brain volume. We are also really curious to look at the regional level. So given sort of what we saw with that total subcortical gray matter volume, we wanted to look at these individual structures deep within the brain individually. So this was a subset analysis of that same cohort that I described earlier. They're very well matched demographically. And again, they completed that physical evaluation, the brain scan, and the neurobehavioral testing. And we used that second tool to quantify percentile scores for these deep subcortical structures, regions like the hippocampus, the thalamus, the caudate. What we saw is that youth with alcohol exposure had smaller regional volumes as well. And these effect sizes were moderate to large in size. So a few examples here, I'm showing caudate and the thalamus, where we can see the alcohol-exposed kids are showing smaller percentile scores for age and sex. And this is notable because these regions have been found to be affected by alcohol exposure in previous research, and have also been shown to be playing really crucial roles in some of these higher level cognitive skills, things like learning and memory, attention, executive functioning. Looking at the relative sensitivity and specificity, we see that slightly higher sensitivity in terms of when we look at small volumes in these regions. We see that that's capturing a relatively higher percent of our participants in the alcohol-exposed group, around 40 to 50%. You'll notice that the specificity is also less compared to that head circumference measurement and those total brain volumes. We think this has to do with something with the fact that in using this tool, we saw a systematic difference where more kids in our control participants sample as well showed smaller than expected brain volumes. And that might have to do with sort of the tool itself. It's pulling data from a number of neuroimaging studies, and even small differences in how that data was processed using that tool can result in differences in some of these values. But even still, we're still able to see these group differences in our alcohol-exposed kids. So together, these data suggest that these deeper subcortical structures might provide additional sort of value in identifying some of these brain-based differences beyond that head circumference measurement that I mentioned. So I'm going to shift gears again and talk a little bit about another tool we're using to look at subtle differences in one specific region in the brain, which is the hippocampus. The hippocampus is a really important structure. It's involved in learning and memory in particular, in addition to other cognitive tasks. And it's been known to be affected by alcohol exposure for many years. Many studies have quantified this. Many studies have looked at how that difference might relate to things like learning and memory. But a lot of that research has evaluated the hippocampus as a total structure, as a total volume within the brain. And now we have tools that can look at really subtle differences in things like the thickness and the curvature and the folding of the hippocampus. So that's what we were kind of curious to look at in this analysis. So this is the same cohort that I mentioned earlier. We applied this tool that basically quantifies things like thickness and the curvature or the gyrification of the hippocampus across the entire surface. And what we see is that kids with alcohol exposure show reduced thickness and reduced folding across wide parts of the hippocampus. So in these figures, the greens and the violets are indicating thinner and less folded tissue in the alcohol exposed group. So we saw these prominent group differences. We also saw relationships with cognitive function. So for example, in the alcohol-exposed group, what we saw was that thinner tissue was associated with lower IQ, lower executive function performance, and lower visual memory. So you can see in these figures here, reds and yellows is indicating a positive correlation between thickness of the hippocampus and performance on these different tasks. So we see that kids who had thicker tissue tended to do better, and vice versa. Interestingly, we saw very minimal correlations with the brain and behavior in the comparison group. So this really might have to do with sort of how alcohol is affecting hippocampal development from an early stage. And we saw less of an effect on that folding measure. So it's really more about the thickness in terms of what we're seeing correlations with cognition. But in general, these data suggest that the prenatal alcohol exposure might affect that development of the hippocampus from a very early stage, and we continue to see these differences even into the school age and adolescent period. So to conclude, we see that these novel tools might be really useful for a number of reasons. They might complement some of our existing approaches and provide additional ways of looking at brain-behavior relationships in this population. In terms of future directions, we're really curious to look at how these tools might be able to sort of tell us about what might be unique to alcohol exposure compared to other neurodevelopmental conditions like autism and ADHD. We're also really curious about how these tools can be used to look at individual level variability in some of the cognitive outcomes that we tend to see. So looking at why some individuals have more challenges versus others, and that might relate to some of these subtle brain differences. And then lastly, as I mentioned earlier, we're curious to look at how these tools can help us evaluate treatment outcomes and why we see benefits for certain cognitive skills from interventions like nutritional supplementation as one example. So I want to thank the University of Minnesota research team for ongoing support with this work. I want to thank the NIAAA for the funding of this study. And I'm excited to take your questions.
Okay. Oh, hi.
Hi.
Yeah, it's a really good talk. Thank you very much. I'm curious. Probably I might miss something in your study. Also, you measured white matter volume, right? But however, in your results, we didn't mention anything about white matter sensitivity because things we already know. Like there are a lot of findings in DTI that white matter volumes or white matter tritography show more changes in children with prenatal exposure?
Yeah, so part of that first analysis, we were looking at volume of white matter across the whole cortex. But you're right, that would be really interesting analysis to look at diffusion-weighted imaging as well. There are normative data sets that can look at that, but we haven't explored that yet.
Yeah, also about the brain charts. Is there like white matter volume brain chart, or maybe the brain chart is all about gray matter volumes?
Can you say that again? I'm sorry.
Yeah, I'm just talking about the brain charts you used. Like,
Yeah.
The brain charts is only about gray matter volumes, or they are both? Like, is there like white matter volumes brain charts?
Yeah, so that first tool I mentioned, brain chart, it does look at white matter volume and gray matter volume in the brain. But what I mentioned earlier is that there is now normative data for diffusion-weighted approaches as well, which would be really interesting to look at, given what you mentioned that we see white matter differences as a really prominent difference in kids with FASD.
Okay, cool. Thank you very much.
Yeah, thank you for the question.
Hi, thanks so much for your talk. I wanted to bring back in the theme of sex differences.
Sure.
And what we've seen in our work with looking at longitudinal brain volume development in younger kids with prenatal alcohol exposure, about 2 to 8 years, is that females with prenatal alcohol exposure have the most reduced volume out of either sex with prenatal alcohol exposure. So I was wondering, if you have any plans to look at sex effects and whether you think that could potentially maybe make a more sensitive tool based on sex.
Yeah, it's a great question. We did sort of explore that with supplemental analyses with this data. We didn't see prominent sex differences. We are limited by our sample size, which is sort of moderate. But it is a really interesting question of whether some of these sex-based differences might play out in typical versus atypical development at this stage as well. So I appreciate your question.
Thanks.
Yeah.
Hi. I have a question about, sorry, I have a piece of paper.
Yeah.
So I know Jeff Wozniak was doing, I think, with the choline studies and the hippocampus, and overall, like improvement on brain function. What exactly does the choline hit? Does it thicken or does it change the shape of the hippocampus? And the second question I have is how much, so you've got a brain alcohol exposed, how much does the brain change in adolescence if somebody is addicted to alcohol, like is drinking and have prenatal alcohol exposure? Does that affect any kinds of shape of the brain or at least function of the brain versus somebody who has never or who has minimal exposure to alcohol in adolescence and adulthood?
Those are really great questions. Yeah, I don't have answers to either one at this point. But very good questions. Yeah, we would like to explore that.
I'm going to ask a quick question. So you have snapshot data from 100 studies, almost 100,000 people. Are you able to look at individual trajectories and to see how they may differ as opposed to what you're seeing in these growth charts? In other words, with a large enough sample to see if there's fall off at certain ages that might distinguish the groups?
Yeah, that's a really good question. So some of our analyses that we've done, not using the brain growth charts, but using other methods, we've been able to show that the developmental trajectory of change over time at this period of age does seem to be atypical in kids with alcohol exposure, and that atypical trajectory of development in something like cortical thickness, as one example, can relate to some of these neurobehavioral differences. It would be really interesting to look at whether these tools might be useful for understanding what we expect the trajectory of development to look like in the typical development, but I don't think people have kind of explored that to date yet.
So this is a little marker, but when HBCD and ABCD are both finished, there will be individual trajectories for 10,000, 11,000 kids in ABCD and 6,000 or 7,000 kids in HBCD going from birth all the way up to age 20, and I think that will be helpful control data.
Yep, exactly.
So I want to thank all of our speakers. I think we're finishing right on time.